Our last topic that we want to cover in this unit, Unit H, is the structure and function of membranes. Now, you may remember that we already covered membrane molecules like the glycerophospholipid that I have shown drawn over here on the right. The key is that membrane molecules are amphipathic. They have a very charged head group and nonpolar tails that we're showing here simply as a cartoon. We can simplify this even further by simply drawing a circle with a negative charge in it for the head group and then making two tails. Now let's say we take these types of cartoon molecules and put them in water. As you can imagine, the nonpolar tails are going to want to flee from the water and congregate together, and that's exactly what they do. You're going to have a nonpolar area that's in some sense reminiscent of a micelle, where the nonpolar tails come together and the polar head groups are interacting with the water. But in this case, we end up having what's called a membrane bilayer. And that's because with two nonpolar tails instead of one, these things tend to spread out and form essentially sheets rather than spheres like we would see with a micelle. And that's what a membrane bilayer is. It's simply glycerophospholipids and sphingolipids that have congregated together in a fashion where the nonpolar tails have come together and they form a sheet in a water. Now, this is my pretty weak artistic skills. Let's go and show you what maybe is a little more realistic. Here's a figure from a textbook. And we can see that this type of cartoon structure is related to a real electron micrograph of a membrane. Membranes aren't really sheets because they can't have edges. These cannot be exposed nonpolar areas. So what happens instead is that they basically curve around, not just in this plane, but they do so in 3 dimensions, and you end up with a sphere with water on the inside and water on the outside. And this is basically how a cell membrane forms, or you can think of it as membranes like for the Golgi apparatus or mitochondria, chloroplasts, things like that. So that's a good way to take a look at what a membrane is like. And what I'd like to talk about at the top here is sort of the physical chemistry properties, in particular of diffusion. And in some sense, this is something that we've already discussed. Going from one side to the other, charge slows down membrane crossing. We use the phrase, a full charge is almost impossible to drag across a membrane. And we can say the same is true for the membrane lipids themselves. If this lipid here was going to try to flip over to the other layer, that's also very slow, because think of the phosphates having to be dragged through that nonpolar area. It's going to go very, very slowly. However, slow is a relative term. And let's do a little exercise here where we consider some flipping rates of various phospholipids and other lipids. Let's consider a cholesterol. Let's consider a phosphatidic acid. And let's consider, lastly, phosphatidylserine. Which one of these would go through the fastest for a flipping motion from one layer to the other, and which would go the slowest? Go ahead and rank those out. Take a pause and see what you come up with. Okay, the idea here is that the less polar the head group is, the faster it's going to go. And what we're going to see is that cholesterol with its little alcohol group is going to go fastest, and phosphatidylserine with its many charges on the phosphate and the serine is going to go slowest, and phosphatidic acid with its phosphate is going to go in between those two. So we'll just call this area rank flipping. flipping from one side to the other. That's a good little way we can connect these variety of molecules we have in membranes to something that's true of their physical properties. Let's go back and consider something like a potassium ion that has to go through the membrane, like in a nerve cell. Being fully positively charged, it's almost impossible to do that. So how do potassium ions get through? Well, let's consider what real membranes are like at the bottom of the page. Maybe we can get this all done in one video. What I'd like to introduce here is what we call the fluid mosaic model of membranes. And if you think about a mosaic in art, what it means, it's like a tile with a bunch of different colored tiles in it. And that's what we're thinking of here too, because membranes are not simple like the way I have them drawn. In fact, they're very complex. They have a lot of different things. They have sphingolipids and glycerophospholipids, and they have cholesterol. We know that. Some of those sphingolipids, like gangliosides, have a lot of sugar structures coming off. So we might have sugars on the surface of membranes. And we might also have proteins. Let's consider a protein that's embedded in the membrane. And I'm just going to show it here having a pore going down through the membrane. Maybe that's the kind of thing that would allow a potassium ion to worm its way through from one side of the membrane to another. This is called a protein channel, or an ion channel, because it lets ions through. Anything that's charged can get through membranes through a channel like that. Now, in general, we call these proteins integral membrane proteins. They're also called transmembrane proteins. And that's because, trans means to cross, and cross the ocean or something like that is a transatlantic voyage. So crossing the membrane fully is going to be a transmembrane protein, and we call these integral membrane proteins because they're fully integrated in the membrane. Now, they might be channels like the one we have here, or they may just be a big chunk sitting in the membrane with no hole going through the membrane. There's a lot of different ways we can do that. We can also have what we call peripheral membrane proteins. Let's call this a peripheral membrane protein. That's a protein that is going to be associated with the periphery of the membrane, and it may be associated by having positive charges like lysines and histidines on its surface to interact with the phosphates on the surface, or maybe it's got donors and acceptors, or positive, negative, that allow it to interact with another protein like an integral membrane protein. The upshot, though, is that these two proteins are very different. We talked about this earlier in our protein function chapter. Integral membrane proteins have nonpolar on the outside, at least in the membrane portion, whereas peripheral membrane proteins tend to have polar on the outside and nonpolar on the inside, more like a traditional water-soluble protein. These are two different ways proteins can be associated with the membrane, and that's probably a good enough introduction to the variety of things we see in a fluid mosaic model of membranes. One thing to finish off, I just want to show you how books can do a much better job with this than I can. Here's a beautiful picture of a membrane. We see integral membrane proteins here. Some of them are just sticking in there. They don't necessarily have a hole going through them. You might have sugars coming off here. The sugars might actually be attached to the proteins. And then down here we have peripheral membrane proteins. So that's a nice view, much better than my artistry. Let's leave it at that.